Yes, I just wanted to encourage everyone for their participation. And again, the code to unmute yourself is *6. And we are recording this call, and all notes will be distributed to the states before posting them on our website. And feel free to participate. Thank you for joining again.
Okay, then let's move on to the updates from the pilot teams. We can start with New York. Michelle or someone else from the New York program?
Hi, yes, can you hear me?
Yes.
This is Michelle from New York. So I have the update for NPS 1, SMA, and Pompeii for you today. NPS 1 and SMA, we began screening on October 1st of last year. For NPS 1, we have screened about 92,000 specimens, and we've had 7 referrals thus far. Those referrals, 2 have been carriers for pathogenic variants and pseudodeficiency allele, and the other 5 have a variant of uncertain significance in pseudodeficiency, so no true cases yet. For SMA, we've had 4 referrals. We had 1 very early in October, and then we had a long dry spell, and 3 within a few weeks after that. So we kind of had the same that other people have had. Our first referral was 3 copies of SMN2. That baby was enrolled in the gene therapy trial. We had the second case had 2 copies of SMN2, and we haven't gotten a follow-up from the care center yet. And these last 3 were pretty recent, in the last few weeks, actually. Number 3 had more than 4 copies of SMN2, so that one is, I believe, entered the SPOT SMA study. And the last one had 2 copies of SMN2, and that was fairly recent, so that baby is still being evaluated. So we're sort of back on track with the number of infants that we're detecting. For Pompe disease, we've screened somewhere around 1 million. We had kind of a lull in December, but we've had 5 new referrals in January, and all of those had 2 variants. We're seeing fewer carriers and pseudodeficiencies now since we've been using Clear, so that's good. We've gotten the phasing from 3 out of the 5 babies, and all those variants are in trans. And interestingly, all 5 of those babies had the late onset splice variant, and one of those babies was a homozygote for that. And we're awaiting the diagnostic enzyme results from the January babies. So that's it from us.
All right, thanks, Michelle.
Sure.
Any questions about Michelle's information?
Hi, Michelle. This is
I have one.
This is Cindy Powell from North Carolina. Michelle, for your protocol, are you doing second-tier sequencing of IDUA gene, or is that?
Yes.
Okay.
Right. Yeah. Right now we're doing that. We're working on getting the GAG assay running.
Okay. Thank you.
Yep.
All right.
This is Kathy Swoboda from Massachusetts. I just want to let you know I was really excited to see the last baby with the 4+ copies and to offer them enrollment in SPOT SMA. But we would love, so that's the LPDR through the NBSTRN. So if there's any other opportunity to refer providers and tell them about that study, if they do not go into a trial, that would be helpful. It's still unclear whether, depending on each trial is unique about whether or not you can refer those babies to go in SPOT if they're in a gene, like in a trial, and it varies by company and what the protocol says. But we could at least enroll and collect the baseline information. So for this 4+ copy baby, we're going to be following that baby very closely. And one option would be to access gene therapy if the baby should present with any symptoms. So I just wanted to let people know that.
So can you clarify, does that accept any baby? So obviously, I guess if it's a 2 copy, they want to get some kind of treatment relatively quickly. So are you saying that any baby that's 4 and greater would enter? What's the timing on that?
No, we enroll every baby we see for LPDR before we enroll them in another trial or simultaneously. And it just depends on the trial whether you can continue to collect. You have to collect you can continue to collect clinical data into the SPOT trial, but some trials preclude you from entering, say, the motor outcome data. You're not going to include the trial data in SPOT until that, that database is going to be maintained until the study is decided or whatever. So you just have to work that out. But we're hoping to get as many babies as possible that are identified via newborn screening. We've enrolled some of the ones from Utah, for instance, as well. So,
Okay.
and of course, every baby we see, whether symptomatic or not in MES.
Okay.
All right. Okay, thank you, Michelle.
Sure thing.
Then let's, glad you made it home. Sounds like everybody on this call, the meeting yesterday.
Yeah.
Let's move on to Wisconsin then. Is May on, or someone from the Wisconsin program? All right, May is still driving back.
Again,
If you're on,
the code
really?
to unmute yourself is *6.
Yep, *6 if anybody from Wisconsin's out there and wants to present their data. All right, then let's move on to Massachusetts. Anyone from the Massachusetts program on?
Ann is on. Hi, can you hear me?
Hello? Gotcha, yep.
So we've been screening by pilot for XALD, MPS1, Pompe, and SMA since last January. We've screened a total of 58,000 babies. For SMA, we have not found any SMA. We had one referral. We did find 5 hybrids by our screen. And as far as we know, through our SMA working group, no babies have come to clinical attention who would have been screened. For XALD, we have 2 confirmed cases, 1 Zellweger, several PBDs, and 3 carriers. I don't have the genetics in front of me. MPS1, we have 2 confirmed cases, 1 possible case. Pompe, again, we have 0 cases, and we're quite aware that our detected cases are out of line with expectations. Our speculation is that Massachusetts may have some reproductive decision-making going on, that because we do know that prenatally council offers screening for SMA and Pompe. But other than that, that's everything that we have to say.
All right, thanks, Ann. Any questions for Massachusetts? All right, then let's move on to Georgia. Is Bill or somebody else from Georgia on? *6 to unmute yourself.
Hi, it's Bill Wilcox.
Hi, Bill. Go ahead.
Hi. So we finished the Pompe MPS1 X-linked ALD a while ago. We're going to be doing a little bit of Pompe and MPS1 shortly using some CDC money, but that's not going to last very long. We're waiting for the state to approve the 4 conditions that are listed there on the slide. And our SMA pilot program is starting in the next day or 2.
All right. Any questions for Bill Wilcox out there? Okay, then let's move on to North Carolina. I guess I heard Cindy Powell.
Yes, I'm on. Unfortunately, I don't have the data from the early check project for SMA. I know we've gotten no referrals yet, here from the RTI group. So I'm assuming no positive screens yet for SMA. And I'm not sure if Jennifer Taylor's on the call and has more information.
Jennifer Taylor's on. Can you guys hear me?
Yes, we can.
Okay, so the update for so SMA is being screened under their early check program, and we have now screened over 1,400 specimens. We still have no screen positives yet. The slide says we're using the Perkin-Elmer assay, which we are not using. We're using the method that was developed at CDC.
All right. Any questions for either folks from North Carolina then? Okay, then.
Mike, it's Joe. I have a question. I'm just wondering, how long has North Carolina been screening for SMA now?
So we started recruitment about the second week of October, and then we started screening specimens October 22nd. So
okay.
What's on the slide is correct, but it's under a consent. Parents have to go to the Early Check website and sign up to have this additional screening done. So far we've only screened about a little over 1,400 specimens.
Okay, I was just curious how the uptake rate may seem to you, as that was kind of interest from a point of view of, you know, how well is it working, but it's a little off topic, I guess.
Yeah, the group is currently working on ways we can improve the uptake rate. Right now, they're sending out letters to parents and asking them to sign up, which is not the most optimal. It's not the most optimal way to recruit families, but it was just to give us a baseline of what our recruitment looks like from just that. And now we're working on different ways, like a social media campaign to reach out to families, to tell them about early check so they can sign up ahead of time.
Yeah, that sounds good. Thank you.
You're welcome.
All right, any other questions? All right, then let's double back and see if anyone has come on from Wisconsin. May Baker or anyone else? *6 to unmute yourself if you're out there. All right, then I guess we shall move on. So in addition
Utah?
to
Utah's not on?
Apparently not.
Okay.
All right, then let's move on then to, you know, we have a group of states we work with fairly closely, and some of the pilots NBSTRN does, but other states have been involved in some of these. So are there any other states out there who are screening for recent additions to the RUSP or candidate conditions for addition to the RUSP, who would like to tell us where they are, what they're up to?
Hi, this is Chris from Virginia.
Yeah, please go ahead.
We just went live for Pompeii and NPS1 screening on the first of the year. And that was with great help from many of the states that have gone before us, including Missouri, Michigan, Minnesota, Massachusetts, New York, Wisconsin, as well as help from CDC and Babies and Perkin-Elmer. We were legislated into bringing up these disorders much more rapidly than our typical regulatory process. So we have elected to go with the Seeker Digital Microfluidics first tier platform, and then our specialists, our geneticists, have requested second tier molecular for both disorders as part of our algorithm. Since going live on the first of the year, we've screened about 9,700 babies. We've started with very conservative first tier cutoffs, and so we've actually reflexed 17 samples for Pompe sequencing or GAA sequencing. 13 of those due to critically low enzyme levels, and then another 4 due to sequential samples coming in under our abnormal or borderline levels. We have received, and I should mention, with great help from Massachusetts, we have been troubleshooting our in-house lab-developed test GAA sequencing assay. And in the interim, we have an MOU with Massachusetts to sequence any of these reflex specimens for GAA. And so they've been working very hard to turn those samples around for us as we continue to troubleshoot. So of the 17 that have reflexed, we've received results for 10. Two samples had pathogenic variants identified. Both were heterozygous and included some variants of unknown significance. And the other 8 specimens for which we've received results have all included variants of unknown or uncertain significance, with an additional 7 samples pending sequencing. For MPS 1, we've reflexed 2 samples to second-tier sequencing based on the first-tier assay. That sequencing has been completed in-house. 1 specimen had a pathogenic variant identified. It was heterozygous, as well as heterozygous for variant of uncertain significance. And then the second specimen is still pending sequencing results. We have not received any clinical diagnosis or further assessment from the specialists. We've only reported the sequencing results out. And per our algorithm, any likely pathogenic or variants of uncertain significance are referred on to the specialists for further diagnostic workup.
All righty, thank you. So does anyone have questions for Virginia? All right, then I'll backtrack one last time for Wisconsin or Utah. Anybody on from either of those states? Okay, then let's move on. Are there any states on who would like to comment on any pilots they're involved in who may not have told us ahead of time? All right. Then I guess we can move on to a new steps update. Is Sika or someone else from that program on? Feels like the week of the government. Oh, yes, we can.
Hi.
Yep.
Okay, sorry, I was on mute. So thank you. We just have a few brief updates. The first is regarding a listserv message that went out through these steps a few weeks ago, or a few days ago rather, for states that are working toward implementation. If there are any technical assistance needs that you have, please let us know. We continue to collaborate with the NBSTRN on activities related to long-term follow-up, but more broadly, if there are TA needs that you have for the implementation of new disorders, there are some resources available, so please reach out to us. And then the second update that we had is unrelated to the disorders, but we just wanted to issue a reminder to this group that for the APHL Newborn Screening and Genetic Testing Symposium that's taking place in April, the awards nomination period is now open, and it's been extended through Friday, February 15th. So if there are any leaders or colleagues in the field that you would like to nominate for one of 4 awards, please go ahead and do that. And if you wanted additional information about those, you can feel free to reach out to me or any of the staff here at New Steps or APHL. That's all I had.
All right, thank you, Sika. Any questions for Sika at APHL? All right, then let's move on to the NBSTRN update. So we're in the process, and we're now in our 11th year of NBSTRN, and so much has changed that we're starting to revisit some of the way we offer blood spots to researchers and others out there in the newborn screening community, and also reevaluating some of our website structures. So we've developed an RFP process, and we're going to have requests for proposals that can be found on our website. And out of that, we'll be selecting contractors, perhaps some that have already worked with NBSTRN, perhaps new ones. But we are, because we're at the very start of a new contract, we're sort of regrouping and making sure everything we've been doing has been working, and are in that stage of redevelopment of our activities. Next slide, is that there? Well, that was the website, but you can see it on the slides. So we're also beginning to regroup around many of our new work groups at NBSTRN. We do try to rotate folks through and are very interested in knowing or hearing from folks that are interested in getting involved in really anything we do. We involve a lot of the grantees who utilize the infrastructure the NBSTRN has developed, but we also have more generic work groups and are always interested to hear from people who are interested in getting more involved. We have one developing now around a tool to collect pilot data. It's going to give us an expanded focus on the pilots, and we really are trying to develop our own data and the way we display our data in a way that's immediately useful to the Advisory Committee on Heritable Disorders of Newborns and Children. Our new consolidated laboratory performance database, you can see in the image the way we're developing it. Sort of obviously the screening steps in the pilots, research activities, implementation of XALD and SCID, the kind of data we're collecting around the numbers screened, the numbers referred, their diagnosis, the ultimate diagnosis of the individual, screening algorithms, true positives target for the target conditions, true positive for other conditions, the cases that are pending. And this is really the kind of data we want to be able to develop into a sort of a continuously updating data display on the NBSTRN website. A lot of people come to us after these calls to find out what sort of the aggregated update is of all the information that we hear on these calls, and we would like to try to make that more available. We're doing another project here that is very much focused on how we approach pilot studies and whether the way we do it now works well or not. And my own sense is that it doesn't work really well because we put things on the RUSP before we start really getting robust data. So we want to be able to build some statistical perspectives of the data. Either we're not sure what it'll be, whether it's going to be confidence intervals around the pilot data based on the number of people who have been screened at that point in time. We know it's a pretty big pilot study till you get to the point where your upper and lower confidence intervals are within 5 or 10% of each other. And there's a pretty extended period of time through 30 or 50,000 babies where the lower confidence limit sits around 0%, which means you don't know a whole lot yet, or at least you're not confident in what you know yet. So our hope is to really be able to provide the kind of data that gives folks a sense of what is the likelihood that the data we're seeing in pilot studies is actually likely to translate out into real practice later, even though there are some aspects of the real world that mean data is usually a bit less robust there than it is in the well-controlled pilot study states. But we do want to try to aggregate things and give a broader view of how all this comes together around those parameters you can see in the bottom right rectangle in this particular slide. So if there's anyone interested in getting involved in any of these activities, feel free to email back to Danielle, myself, or Amy Brower about your interest in this particular activity that we're developing or others that we're involved in. Next one up then, please, Danielle. All right. Information around some of our data collection efforts. As I said, we're really trying to collect and disseminate the data from the pilots to try to facilitate implementation And as our data gets more and more robust, various of the programs, CLEAR and others, begin to become discovery vehicles for finding other associations that may be informative for likelihood of true positives or even false positives and those sorts of parameters. We would like to be able to inform new steps and others. We work increasingly closely with new steps as NBSTRN moves its way through pilots, and new steps then begins to implement in states that are in sort of the second wave, where it's really more about analytical validity and trying to replicate the levels of analytical performance that the initial states had to realize the clinical data that they were able to capture. So we are trying to partner up with other ongoing activities in newborn screening, so we all have some synergy around the kinds of work we're doing. Some of the initial focus of our data collection has been obviously the NICHD pilots that you've been hearing about on this call. Some of the pilots done through the North Carolina IQ pilots, where we work with under task orders to bring additional states into the pilot studies. It's my understanding that that will be recompeting probably in I think it's about a year, within the next year, that states that may not have chosen to become involved initially will have another opportunity to apply to be among the states that could be approached to participate in the studies that NICHD funds. And then we're also looking at obviously some of the expanded areas that may not be conditions on the RUSP, but are very interested in sort of new conditions that may be earlier stages and not have gone to the advisory committee. We're actually going to be publishing a paper, and then probably around in about 2 months, where we've taken a pretty comprehensive look at the pipeline of conditions that are coming up, that are becoming candidates for newborn screening. There's a fair number already, probably at least a half a dozen, where we already have the analytical data because they're amino acids or other analytes that we already capture in tandem mass spec. That would actually allow some of the conditions. Proximal urea cycles are an example of a group of conditions where we have the analytes already, and they're already part of a platform that's run for other conditions. But what we really need is a more virtual kind of pilot that gets at the long-term follow-up side and takes advantage of the literature that's been coming out on some of those conditions, and looking at alternatives to sort of purely prospective pilots for some of the conditions where we already have a lot of data that might be used retrospectively, and certainly pilots that have already taken place in states like California and Minnesota and others. Next slide, please. All right. So these data collection efforts are really much like we're doing now. We try to report back on the monthly calls. We get data from states by email that we're able to at least present to you through the slides. At the start, we want to be able to develop the capacity for direct uploads through this new data collection effort that we're developing based on REDCap surveys. REDCap being a platform that's fundamentally an Excel spreadsheet, but it's something that is fairly easy to use in many states and research programs, and accepts uploads of data. But we really do want to get much more comprehensive. It's very clear from our work looking at the sort of data requirements of rare diseases at the population level, where we know so little because we're so biased by the sick people who present for care, and increasingly appreciate the fact that once you get to these screening programs, that the breadth of severity, certainly on the mild end, expands tremendously. Our perceptions of penetrance begin to change an awful lot. So we do want to be able to capture much more robust data, and our feeling is that goes well beyond the period of the RUSP. Certain analytical validity is a fairly straightforward statistical concept, and depending on the incidence of the condition, may be a fairly straightforward number of cases that are needed or number of individuals in a population. But clinical validity is becoming an increasing problem as we have much more variability in the diseases that are identified, so that when you've seen one, you've seen one. And we need a much broader perception of what's coming out of the programs to understand some of the clinical validity issues. And increasingly are appreciating the fact that it's only often a subset of a disease that we're able to actually take through the entire pilot to get to the point where we have outcome data that is really what the pilot's all about. And that there's other later onset forms or milder forms that may not go to treatment, where they may not be the primary targets of screening. And we just came off a 2-day meeting where a lot of those kind of ideas were discussed, and a lot of discussion about what is the target of screening and how can we minimize some of the off-target things like carriers and such. But that's an ongoing effort that will continue through the Federal Advisory Committee. Next slide, please. All right, so we're back to soliciting recruits to our activities. Anybody interested in helping us develop these data collection systems so we can bring more of it online? And as more and more states get involved, we have less time on these calls. So we're very interested in developing much more efficient systems and comprehensive systems for showing data at a national level. If you're interested, feel free to get back in touch with us. Next slide, please, Danielle. So our conference call schedule, first Thursday of each month. If you need to contact us, there's Amy Brower's and my email addresses. If you want to present at a future meeting, get in touch with us. If you have interest in participating, you can get to us through those emails. If you have thoughts of other things we ought to cover, we're wide open to suggestions and to adjusting to the needs of the broader community. So feel free to be communicating with us around things that you might think would be of value to you and others on the call. So I'll open it up to any general discussion or comments or questions.
Mike, this is
Yes.
Bill Wilcox. I haven't been able to be at any meetings in a while, but what's the process going to be for deciding what kinds of disorders are put out on the task orders for the IDIQ?
That has always been a decision that is at the NIH, NICHD. Looks at things that get presented on calls and at other meetings where they do hear about candidate conditions. They try to look at where we might be able to multiplex, for instance, because the more conditions we might do in a single pilot, the better. But it's really a decision that NICHD makes as to what the next pilot's going to be. The only thing I'm aware of is that they intend to announce one sometime in the very near future. I'm not certain exactly what it'll be. We've had a few discussions, but it's not within my purview to decide what they're going to opportunities for.
Yeah, it just seems to me that there should be some kind of organized process to help advise the NICHD rather than them just having to pick it themselves.
Yeah. Yeah, I hear you. As I said, they certainly are aware of the data we've been collecting. We're actually going to be sending out a survey probably the first of next week that lists it must be a list of maybe 30 conditions where we're aware of the potential for a test based on particular analytes or other kinds of, it's not always metabolic, so it's not always analytes. But there's a fair number of conditions where low levels actually could be informative about a potential condition. The proximal urea cycles, many people are already beginning to include that in some of their screening because it's coming out and some of the analytes are in the amino acids parts of the tandem mass spec output. We could, I'm happy, we could make that a part of this call if people think there are really priority areas that would be a good focus for pilots. We're happy to hear about them. We are a contractor for NICHD, so we do meet monthly and are able to communicate things that we hear through these calls. I know they're often on these calls. So yeah, maybe that's a topic we could bring to this call, because the pressures are often different in states as to both why someone might be recommending something be added or pressuring a program. But it's always good to know where there is interest and where there might be political pressure to start pilots of different kinds. Do you think that would be useful to try to include a discussion of that type in this call?
Yeah, it's kind of this is a rapid fire forum, so it doesn't lend itself to a lot of discussions. There is an organized group that considers applications for inclusion on the RUSP, but it doesn't seem like they've taken on their purview to consider, things that might be included on it, but they need pilot data in order to consider it.
Yeah, I hear you. I mean, Melissa Parisi at NICHD is a member of the advisory committee representing NIH, so she certainly gets exposed to what's happening at those meetings and what the committee's considering and what, any discussions that come before them about potential pilots. But our list is quite long. There's actually a very long list of conditions for which there is probably already a screening test available. And there's a fair number with treatments available. I've been looking at the clinical trials world fairly carefully, and there's a pretty long and daunting list of new therapeutics in stages or phase 2 and 3 of clinical trials.
I know, I love it.
Yeah, well, many of them are going to be quite challenging. If you think about a condition like Duchenne muscular dystrophy, for which some pilots are about to launch, the treatment that's out there outside of steroids that might be applicable to everyone, exon 51 skipping, which is the drug that Sarepta has, really targets about 15 to 20% of the patient population likely to benefit from that treatment. And it really starts to change your perceptions of how many patients you need in a clinical trial in order to test whether or not the treatment gets you the outcome you expected, when only 15, 20% of all the patients identified in screening are going to be candidates for the treatment. It's the kind of thing that makes you begin to think about what is the primary target of screening. So yeah, we'll see what the next, as I said, I think NICHD is planning on another announcement of a task order for another set of pilots. After that, I think we can begin to look hard. We're much more worried about the capacity, national capacity, for the number of pilots that are out there potentially to be run, given that so many of the questions that have to be answered can only be answered in the context of a general population kind of pilot study, where we get away from the biased ascertainment of thinking we know what the disease is based on the sick people that come for care. So yeah, I think the next year, certainly for NBSTRN, will include a lot of activity to try to get a more coordinated system among the various grantees and programs really that are involved in different aspects of newborn screening, and just try to improve our communication and minimize the duplication of efforts that all of us welcome, so that if somebody's doing something well, others of us don't try to replicate it, and we just work more closely together. All right. Any other comments or questions, ideas of things we might discuss on these calls? All right, then I guess I get to
excuse me, Dr. Watson.
award you all 20 minutes. I'm sorry, go ahead.
Yes, I see that Dr. Baker is online. I'm wondering if you would like to give an update for Wisconsin.
Oh, for Wisconsin?
Can you hear me?
We can hear you now.
We can.
Oh, okay. Yeah, because I keep trying to say something. Wisconsin update is we are still waiting for the state in terms of for the rulemaking allowed us to do the SMA. But our Pompeii pilot come to the end. The plan is end March. So this is two different systems. One is a pilot study, and another one is a state routine newborn screening. And we will have a gap, and I don't know how long. And another thing is interesting.
I don't know, I can't say too much yet, is a lot of discussion in our state. And we really, for us, we screening about 80,000 only we identified is a potentially later onset. So we are trying to refine the term, and we still feel strongly infantile should be screening, but if we do keeping report a dozen, later onset, what we are really doing. So we are discussing this. We start use the term newborn screening for infantile Pompe. So that's, we don't know at the stage. And because again, our state have the similar, like a federal, we will have a nomination process. And I don't know after all this, a dozen later onset identified with infantile, I'm not so sure how exciting our metabolic physicians want the nomination or not. So remain to see. And I do have a question for Mike. And could you just specify, you said they have an upcoming announcement from NICHD. Is an announcement for the disease for the pilot study for current grantees, or they will have a new cycle grantees? I don't know because, okay, thank you.
So, yeah, so the IDIQ is the structure they developed to be able to manage pilots more efficiently. As you probably know, if NICHD was to decide to do a specific pilot, before they had this other IDIQ system, they had almost a year and a half of sending the proposal downtown in Washington to get higher level approval to put out a grant offering for that particular pilot. So they developed the IDIQ program as one in which they funded a central entity, which I believe is Don Bailey right now at RTI. And through that mechanism, they're able to supplement that particular contract with task orders that can be done much more rapidly. So the task orders thus far have been for XALD, SMA, MPS1. They've been doing them typically in December and January, make an announcement of what the next task order will be for the states that have already been approved to be involved in that particular program. Mm-hmm. And so it's one of those task orders that is likely to be announced in the next several weeks.
Okay, thank you.
You're welcome. Other questions or comments?
Again, to unmute yourself, it is *6.
All right. In the absence of anybody coming on, then I will award back to you 15 minutes of your life. Thanks for joining us. We appreciate it.
Thank you all for joining today's Newborn
Screening
Pilot.